Hello students, I'm Dr. Arpita here. Today we will be talking about Apomixis. This is a very important part of your embryology syllabus. So let's get started. The word Apomixis is derived from two words. So it's a combination of two words. One is apo, the other is mixis. Apo means without and mixis means to mix or mingle. Right? The term was coined by Winkler in the year 1908. Okay? Apomixis is the phenomenon of embryo development without gametic fusion and meiosis. It means what? Embryo normally develops after the fusion of male and female gametes, right? But here you see embryo is developing without the fusion of male and female gametes, and meiosis is also not taking place here. Thus it is also called apogamy, right? It may include any form of vegetative reproduction, right? Now you see this flow chart. Apomixis can be categorized into these types. The first type is non-recurrent apomixis. Second is recurrent apomixis. Third is adventive embryony or sporophytic budding. And fourth one is change of flower to vegetative reproductive bodies. Okay? So let's get started. Non-recurrent apomixis. What is that? Embryos are formed without fertilization. Okay? That means what? The male cell is not fertilizing the female cell, but embryo is developing or embryo is forming. Okay? They are haploid and sterile. These are two very important points you should note. They are haploid and sterile. If the male cell would have fertilized the female cell, then it would have resulted into a diploid embryo, which is 2N. N means it is haploid, means like this, it is N, and it is sterile, right? The haploid embryo is formed from the egg or any cell of the female gametophyte. Okay? Now see, when the embryo is developed from the egg without fertilization, the process is called haploid parthenogenesis. So what is haploid parthenogenesis? It is the phenomenon of the development of embryo from egg without fertilization. Without fertilization, please note. Okay? And they contain only a single set of chromosomes. Only a single set of chromosomes are there. It is found to occur in Solanum nigrum, Listera ovata, Orchis maculata, all these plants, right? They cannot repeat another generation since they are sterile. We just now talked about this. They are sterile, so they cannot continue the generation, and they are thus known as non-recurrent. Had they been fertile, it would have been called recurrent. Since they are sterile, it is called non-recurrent. Okay? When the embryo is developed from any cell other than the egg of the embryo sac, it is called haploid apomixis. What is it called? Haploid apomixis. So haploid apomixis gives rise to an embryo that is formed from any other cell but not the egg. Okay? It is either called haploid apomixis or haploid apogamy. Right? The embryo formed is haploid and may be formed from a synergid cell or an antipodal cell. It can develop from either a synergid cell or an antipodal cell. Okay. When the embryo develops from male nucleus, it is called androgenic haploids. It is found in Nicotiana. So what is androgenic haploid? When the embryo is developing from the male nucleus. Here too the plant is sterile. So in all cases you see the plant is sterile. That is why it is called non-recurrent. It cannot continue the generation. Okay. According to Dr. Maheshwari, the haploid embryos are formed owing to one of the following reasons. So there are certain reasons behind the development of these haploid embryo. What are these? First is absence of pollen tube. If the pollen tube is absent, then haploid embryos can result. Next is what? Inability of the pollen tube to discharge its content. First is there is no pollen tube. In the second case, see, the pollen tube is there, but it is not being able to discharge its content. So it is as good as being absent, isn't it? Third is insufficient attraction between male and female nuclei. There should be some amount of attraction between the two gametes, isn't it, for fertilization to occur. Next is what? Early degeneration of male nuclei. If the male nuclei degenerates before the maturation or before the female cells are ready, then what is the point of having that? So in this case, this type of case also results into haploid embryos. And last is what? Discordance in the time of maturation of the egg and entrance of the sperm. So there is no synchrony. There is a discordance in the time of maturation. The time of maturation is different. This also results into haploid embryos. Right? Next we come to recurrent apomixis. What is that? The embryo is formed without fertilization and are diploid and fertile. Over there, what did we see? In non-recurrent, haploid and sterile. Here in recurrent apomixis, it is diploid and fertile. That is, they are capable of reproduction. That is the reason they are called recurrent. It can recur. It can repeat. Fine. The embryo sac may arise either from a cell of the archisporium or from a cell of the nucellus. Okay. See, there is no reproduction in number of chromosomes, and all the nuclei of the embryo sac are diploid. That means 2n. See, production of diploid embryo from a cell of the sporophyte without meiosis is called aposory. So, if you have a question like this, define aposory, what will you say? It is the phenomenon of production of diploid embryo. Diploid embryo, please note this, from a cell of the sporophyte without meiosis. Meiosis is not taking place. It is called aposory. Right? The production of diploid embryo from an archisporial cell without meiosis is called generative aposory. Okay, from an archisporial cell. Note this, from an archisporial cell. It is called generative aposory and is found in Parthenium, Eupatorium, etc. Right? Next, what do we have? See, the production of diploid embryo from a cell of the nucellus. Nucellus without meiosis. Obviously, meiosis is not happening, so without meiosis is common to all. You will have to remember these terms. Cell of sporophyte, then it is called aposory. From archisporium, it is called generative aposory. Now, if it is developing from the cell of nucellus, it is called somatic aposory. It is found in Hyraceae. Right? See, diploid embryos are formed in 3 ways. What are they? Diploid parthenogenesis, diploid apogamy, and somatic aposory. Right? Now we'll discuss about each of these. See, diploid parthenogenesis. Embryo develops from diploid cell, that is diploid egg cell, without fertilization. It occurs in Taraxacum, Antenaria. Here, no reduction division, that is meiosis, is observed during egg formation. Their egg cell is diploid. Obviously, no reduction division is occurring, therefore it is diploid. This unfertilized egg gives rise to a diploid embryo. Very simple. Next is what? Diploid apogamy. Embryo is formed in diploid embryo sac from any cell other than the egg. It can be any cell, from the synergids, from the antipodal cells, but not the egg, without fusion of male nucleus. Right? The embryos formed are diploid and they are found in Alea, Malcamia, all these type of plants. And then we have somatic aposory. In somatic aposory, let's see what is there. The embryo sac develops from the cells of the nucellus. Okay. When it is nucellus, it is always called somatic. See here. See. If the embryo sac develops from a cell of Archisporium, it is called generative aposory. It is found in Eupatorium. Alright. Next, what do we have? We have adventive embryony. See, embryos arise from the cells of the nucellus or integument. Fine. The embryo does not arise from the cell of the gametophyte. The embryo is developed directly from sporophytic cell by budding. Very, very important. So, from where is it developing? The embryo is developed directly from a sporophytic cell by budding. Okay, the sporophytic cell are densely protoplasmic and divides actively to form a small mass of meristematic cell. This small mass of meristematic cell is quite capable of rapid division. Okay, and it later forms, differentiates, divides and differentiates to form the embryo. Okay, this is found in citrus, trifoliata, ficus, mangifera, all these plants. Right? Next, see, change of flower into vegetative reproductive bodies. Here, the flowers are replaced by bulbils. Okay, these propagules are formed by the sporophyte only. On germination, these bulbils give rise to new sporophytic plants. So the bulbils directly germinate into, or they grow into new plants. Okay. Next, what do we have? 3 types of bulbil formation. The propagules are formed outside the floral regions, right? And despite the occurrence of functioning sex organs, no fertilization or seed setting take place. Okay, this is found in agave, elodia, all these. The next is what? See, the propagules are formed outside the floral regions and the plants are sterile. Here the plants are sterile. Okay, it is found in Fritillaria, Lilium, etc. They propagate by bulbils or bulblets. Okay. Third, the propagules are formed on the floral branches either in addition to the flowers or in their place. This is called vivipary and is found in allium, agave, etc. Finally, we should know what is the significance of this. What are the significance here? Occurrence of haploid parthenogenesis is of considerable value in genetics. Okay. Why it is so significant? See, true breeding homozygous types are obtained. Next, indefinite multiplication of favorable biotypes without any variation. Since there is no mixing, crossing over, there is no variation, so you can indefinitely multiply the favorable biotypes, right? When there is a crossing over, there are chances that you get new combination, variations are there. But since there is no crossing over, variations are also not there, so you can indefinitely go on multiplying the favorable biotypes, right? Third is what? Apomictic genera contain high chromosome numbers. And finally, by apomixis, a particular race can be retained. You can retain a particular race because you can again refer to this point. There is no mixing, no crossing over, no variation, so you can retain a particular race. And finally, apomixis is of great value from the origin of species point of view. Important apomictic crops are citrus, mango, blackberry, all these. So apomixis is really very important for us to study plants as a whole. We should know what is happening when there is fusion of two gametes, when there is no fusion of two gametes. Okay, I hope you have enjoyed this lecture. In case you have any confusion, please feel free to get back to me. I will try my best to help you out. Okay, so stay connected for our next videos. Till then,